Everybody, we're about to get started. I want to welcome you today to our webinar, the last webinar in our series, if you've been joining us each week. Before we get started, I'd like to cover a couple of housekeeping items. We will be using the question and answer box for any questions that you might have during the presentation. If you have any issues with your technology, audio or video, please use the chat box. If you have a question and you raise your hand, we're unable to contact you individually. So again, please use either the question and answer box or the chat window. So another item, we are recording today, and if you'd like to revisit any of the content, that information will be available on our training site, hopefully in the next couple of days. So let's get started. Joining us today to talk about sudden oak death and Remorum blight are Rachel Bomberger and Marianne Elliott from Washington State University. And I'm going to turn the floor over to Rachel and Marianne, and they'll give you a little bit of background about themselves before we get started. So thanks for joining us.
Thank you, Rachel. Welcome everyone today to our presentation. My name is Rachel Bomberger. I run the WSU Plant Pest Diagnostic Clinic over in Pullman, Washington, which is functionally Idaho in many ways. So there I work on a lot of different issues, primarily crop problems, but on occasion I do come across landscape as well as first detector issues. Jenny Glass is my counterpart over in Puyallup, Washington, which is Western Washington. And she is one of those that primarily deals with a lot of landscape issues and is quite involved with Remorum, leaf blight, and shoot blight. Marianne?
Yeah, excuse me, I'm Marianne, and I am a researcher over in Puyallup. So I work with, anytime Jenny gets a Phytophthora in the plant clinic, she shares that with me. And I've been working on P. Remorum basically since the beginning, so I'm very familiar with it. So thanks for coming. Go ahead.
All right, so Marianne will be our expert. I will just be the talking head for this presentation. So to get started, first of all, the Npdn, the National Plant Diagnostic Network, divides the country and surrounding areas into 5 different regions. So we're part of the Western Plant Diagnostic Network. We're up here with our little cougar logo. So I'm about here, nearly in Idaho, and then Jenny and Marianne are right over here. So the National Plant Diagnostic Network, the Npdn, mission and role of its first detectors. The Npdn mission is to enhance national agricultural security and to limit the impact of endemic, emerging, and exotic pathogens and pests on plants in the United States. How this is done is first through early detection. This is the critical step when it comes to actually mitigating the impact that these pathogens and pests have. Now, early detection is what we rely on first detectors for. So first detectors tend to be folks in the green industry, so maybe landscaping, nurseries, public gardens, forestry, as well as folks like master gardeners, anyone who sees plants in their day-to-day activities, even hikers and folks who just love the outdoors, are critical for this early detection and first detector point. Next comes accurate diagnosis. This is where people like myself, Jenny, and Marianne come in. We're the ones who receive samples that are suspect from our first detectors, and we go about the process of figuring out what pathogen or pest is actually infecting the plant. Sometimes, actually most likely, it's a native or an endemic organism, but on occasions, we do find those exotic pathogens and pests. And when that happens, we start this waterfall or chain of communication. The diagnostic clinics then work with their state departments of ag. The State Department of Ag might test that sample, also determine that it is a pathogen or pest of concern. And that's when USDA APHIS, the Animal Plant Health Inspection Service, comes in, and we all start working together to try and mitigate the impact of this pathogen. This is rapid response. But the biggest thing to keep in mind is without early detection, there is no rapid response. So this is why first detectors are so critical to the mission of the National Plant Diagnostic Network. So in places like Washington State, where we are a major port area, and as well as a state that relies heavily on its green industry and agriculture and timberlands as for wood materials, food, landscape plants, as well as recreation, they have a vested interest in staying on top of these invasive species. So your state might also have a first detector program. Ours is partnered with Npdn. We do similar trainings, but sometimes your state-based program might have a broader scope. For us, there's a lot of focus on terrestrial invasive plant species. We also work on terrestrial animal species. Feral hogs never fail to inspire a lot of enthusiasm at talks, as well as pests of different sorts, including Asian long-horned beetle, and then quagga mussels for us. Because again, we have a lot of waterways that are important for our recreation and hydroelectricity. So your state might also have a sister program. So as we get started to actually dive into tanoak death or Remorum blight, one of the first things we have to talk about, and I'm sure every plant disease talk you might have listened to brings up a slide similar to this. This is the plant disease triangle. This is our recipe for disease. We have 3 ingredients that are critical. It's kind of like when you're making grilled cheese, you could add in a lot of different elements, but you still need the bread, the cheese, and heat. So this is our underlying 3. We need a host that is susceptible, so that is our plant, a pathogen, in this case it's Phytophthoramorum, and the third and most critical part, depending on if you're a cheese or bread person for grilled cheese, is the environment. We have to have an environment that allows this process, this cycle of infection to occur. When we talk about Phytophthora, this is even more critical. So with Phytophthora, it's important to keep in mind that this is not a true fungus. It is an omycete. It is this weird, I guess weird wasn't the right term, especially since Marianne's here, I can't call it weird. It is a unique organism that's not quite like a fungus. It's more like a water mold. So we kind of lump it in with fungi, but it's important to know that they are different, especially when it comes down to management. We have different management for this particular group of pathogens. So Phytophthora genera in general, pythiums, and downy mildews. They actually require a totally different spectrum of fungicides. We even still call them fungicides, as well as they have different requirements for their disease and life cycle. For this group, that primarily has to deal with water. So these organisms often require water as part of their life cycle or disease cycle. And because of that, we actually use that sometimes to our advantage when we're trying to manage this particular pathogen. And I don't know how many of you actually have the ability to speak Greek, but if you did, you would notice that the word phyto means plant and thora is destroyer. So you get the general impression of what plant pathologists thought of this pathogen when we first found it. We're like, that thing kills plants, we should definitely keep our eye out on it. It's important to keep in mind that not all Phytophthora species are the same. We have many native and endemic species, and when we say endemic, that means pathogens or organisms that have been present for so long that we kind of consider them part of our natural ecosystem or our natural environment. Phytophthora is also a pathogen on a lot of different crops, primarily a lot of vegetables, fruit crops. I get a lot of apples and cherries with Phytophthora-based diseases. It's common in our landscapes and more. Probably every different plant system you can think of, there's probably a Phytophthora that's involved with it. Phytophthora is even more famous than tanoak death for the Irish potato famine. This involved the late blight pathogen Phytophthora infestans. So one fun thing, and again, I say fun because I'm a pathologist, one fun thing about Phytophthora is it actually, here's the zoospore down here, it has this specialized spore that actually allows it to kind of swim or waddle towards plant roots. And that way it's able to initiate infection. We'll talk about that a bit more. So Remorum blight, aka tanoak death, as a disease. So again, it's a water-dependent organism. In order for it to successfully complete its disease and life cycle, it relies on water. Its native range is unknown. However, It has been found in streams in Vietnam, so there is believed to be a possible East Asia origin to this organism. Keep in mind there are many native Phytophthora species, so just because we find a Phytophthora doesn't always mean that it is Remorum. There are at least 31 species or genera of confirmed hosts, possibly more, and we'll talk about there's a difference between a confirmed host and an artificial host. So a confirmed host is one that we find disease naturally starts happening, the progression happens rapidly, and we can find it happening in different environments. With an artificial infection or an artificial host, that means there's a particular environment and set of circumstances that have allowed the pathogen and this disease to progress in infecting that host. This particular disease can cause kind of two sets of symptoms. That's why we sometimes call it sun-noak death or Remora blight. So it causes bark cankers that may girdle and eventually kill the host, or foliar blights that might be a reservoir. So those are the ones that are affecting the leaves, the shoots, kind of the upper canopy of a particular plant. And it actually helps spread the disease, especially in a forest-based system. So the biology of sudden oak death, it's going to thrive in cool and damp climates. So it loves the Pacific Northwest, particularly the west side of the Pacific Northwest, where we have a lot of cool and damp temperatures and situations. Naturally vulnerable species include tanoak and oak species, Quercus species, as well as maple, larch, and more. So depending on the particular makeup of your forest, your environment, you might see different species impacted significantly. I know in California, it's primarily the tan oak forests that are impacted, and I believe over in Europe, it's larch that is frequently targeted. For us, primarily in the US, and especially like PNW area, our top 5 hosts, as Marianne calls them, are filthy 5, are rhododendron species, so that is rhododendrons and azaleas, pyrus, viburnums, Camellia, also known as mountain laurel, and Camellia species. These are the ones we're most likely going to find having the infections. They tend to be a preferred host of this pathogen. There are many more artificially susceptible. I think last time I checked, there was either 95 or 130 artificially susceptible species. These are ones that during inspections, researchers, State Department of Ags, or APHIS has found Phytophthora Remorum associated with that plant, maybe causing a light infection, but not necessarily killing or significantly impacting that species. That's what we call an artificially susceptible, where somehow it's kind of like, have you ever heard of people who made grilled cheese in their toaster? Kind of like that. Not really the right way to make the disease, but it did happen. I'm obviously hungry, sorry everyone. So keep in mind that depending on how the Phytophthora comes in contact with the plant, what different tissues the Phytophthoramorum comes in contact will change the biology, and the host will also dictate how symptoms are going to present. So it can infect different areas of the plant depending on species. For our sudden oak death, this usually means it's infecting through the trunk, through the roots, and moving up to the vascular tissue. This can impact branches, as we'll see with flagging coming on, or it can impact the leaves and twigs, often with the spores landing onto the leaf and then moving into the twig tissue. So tan oak and oak species in particular are often killed in the US. So identification. One thing to note, this is one of those, I'm sure as you've listened to this series, you've realized that identification is usually the tricky part. I don't know why exotics are so good at mirroring and copying pathogens or abiotic problems that we typically see, but they can be extremely difficult to distinguish from other plant diseases and disorders. So here we have a rhododendron showing some leaf spots. Now this is a rhododendron that does have sudden oak death or Remorum blight, so we see discoloration at the tip of the leaf. Have any of you ever seen a rhododendron that does not have spots? Because I haven't. I find rhododendrons covered with spots all the time. So sometimes they're actually other diseases, sometimes they're just disorders. They're sunburns, they're damage that has happened. So it can be extremely difficult to ID. Even when we get those very shocking symptoms where we know something is absolutely not right, like these bleeding and rusty cankers that you can see on the sudden oak death style diseases, and you cut in, you can see this dark delineation of vascular tissue. Even then, we have other pathogens and pests that create the same symptoms. Because of this, molecular diagnostic testing is required to ID. So most of the time, a diagnostician will find Phytophthora, and if it's one of those hosts, we go, we're gonna have to dig into this a little bit deeper, see if the morphology of that particular Phytophthora is starting to look a little Remorum-ish. And when that happens, I send a panic phone call to Marianne saying, I need to send you something. But that's where we move forward to molecular diagnostic testing. So we actually go down to the DNA level to determine if it is indeed Remorum, or maybe it's just a native Phytophthora, an endemic Phytophthora. Keep in mind, we'll have two different sets of major symptoms, the canker or the leaf lesions and shoot diebacks. So with the canker, sometimes you can see this dark red-brown bleeding liquid, which in some ways is a really nice sign or symptom, because you go, well, that's in no way okay. I need to check this tree out a little bit more. And then under that bark, you can see dark brown to red patch discoloration. And keep in mind, some species might have some red cambium layers associated with them, but you'll see a dark discoloration. That's what we're looking at right here. We can see where it's been infected. There's a sharp line between healthy tissue and infected tissue. Now with leaf lesions, we tend to see the lesion developing near the tip of the leaf or along the midvein. That is because that's where water tends to collect and tend to pool, which actually allows the Remorum blight pathogen to initiate infection. With shoot dieback, we tend to see these terminal ends begin to wilt, droop, maybe there's some leaf drop, and then eventually death of those terminal shoots. So with the foliar damage, our foliar hosts are often understory shrubs and woody ornamentals. So understory shrubs if they're actually in a forest environment, but for many of the times that we might find Remorum shoot and leaf blight, it's going to be a woody ornamental that could be in someone's landscape. Again, these are Camellia, Rhododendron, Viburnum, Pyrrhus, Camellia, but also include things like Magnolia, Leucanthia, I can't say that word ever, Osmanthus, and Syringa, so lilacs. These are the ones that will get that leaf blight, defoliation, and shoot dieback. So again, here are some images of those lesions on the end, especially associated with the midvein, tip lesion at the very end again. And here's some shoot dieback. So shoot dieback can be one of those symptoms that will really flag you down because you see this wilting, sad shoot, and it does tend to clue you in that something's not quite right. So here we have a Pyrrhus with infection. Again, this is one of our filthy 5. So again, major discoloration along the midvein. So if you happen to be inspecting, so I think this photo shows this well. So here we see some discoloration on the top side, but when you flip that leaf over, you'll see a lot more discoloration on the underside, particularly associated with that midvein. We also see some dieback of this terminal shoot tissue. This is Viburnum, where we have leaf spots, possibly some discoloration going on. But given these tough, leathery leaves, we often also see sunburn. So sunburn can look very similar to this as well. Here is Camellia. Again, more leaf spots. One thing that I like to note about this photo, these are beautiful photos by Marianne. This gives kind of a wet impression. I think one thing to notice with these Remorum blights is they can give on almost a water-soaked appearance, or I think a little bit more watercolor-like symptomology in their lesions, where we have this browner, drier, papery discoloration, and then it's surrounded by a more mottled, or just like I said, it almost looks like someone's watercoloring a lesion there, discoloration associated with it. We can also see that nice tip area blight along these as well. And when I say nice, keep in mind, I am a pathologist. I can't help but like how these pathogens function. Next is a rhododendron. Again, we see drooping, wilting, along with tip blight. So this rhododendron potentially is root system could also be infested. But then other parts of the plants look great. And then Camellia. I definitely would look at this and go, well, that poor plant just looks like it's struggling, but it happens to have sudden oak death, Remorum blight associated with it. So sudden oak death, when we have stem infections, these tend to be our tree species. So beech, dogwood, true fir, oaks, Doug firs. Keep in mind with conifers, you'll see more of a needle blight and dieback. The roots get infested, and typically this happens, especially in a natural setting where those understory plants, our filthy 5, might have those shoot infections. That moves, those spores move down either with rain or just fall naturally into the soil with the leaf litter. And then from there, the soil gets wet, that organism, that zoospore, the sporangia, move down into the soil profile, where once there's been enough rain, it's actually able to smell its way to the roots and initiate the infection. So it spreads to the roots, then up through the crown and stem, resulting in stem lesions like we can see here, this red-brown rusty discoloration. As disease progresses, we eventually start to see flagging. Now, flagging is the symptom where one limb discolors or droops, or you notice symptoms on one limb. In many ways, this is a really helpful symptom because it indicates that something out of the ordinary is going on. When we see flagging, it tends to let us know that there's a vascular interruption, not in the same way that drought stress happens where all of the top branches start to be affected. With flagging, just one branch starts to turn color. I like to think about it as the plant is literally flagging you down to say something's not right here. I need to be examined. Remember, because plants can't talk, so they have to clue us in with their symptoms. This will then progress to dieback as more and more of the plant begins to die and decline. And then you can find those bleeding cankers or rust-colored cankers, and then eventually death, right? Sudden oak death is in its name. It is its scary name. We were talking before about how murder hornets are a more popular name for the Asian giant hornet than saying that. So, but sudden oak death does come by its name earnestly. Keep in mind, especially with these tree species, that the host response will vary based on how that very special interaction between pathogen and host does proceed. I just find this terrifying. So here we have an example of the rust-colored spots along with that bleeding canker. To me, this is so graphic, and there's something visceral about seeing something bleeding, especially a tree, which we really don't think should be bleeding. And then unfortunately, underneath that tissue is an immense amount of rot to get to the point where the stem is actually discolored and producing that ooze. We know that that infection has proceeded quite significantly. So the last part, but honestly the most critical part of our disease triangle and understanding how sudden oak death and Remora and blight happen is the environment. So this particular pathogen requires water for its infection cycle and completion of its life cycle. Its spores are smart enough to stay dormant. So that means they're just like a volcano. They're just hanging out, waiting until the right environmental conditions. And for this pathogen, that means wet. So it needs, I believe the last time I read literature, it's 4 hours at soil saturation, so 4 hours of enough moisture to trigger spore germination. And because it's not a stupid pathogen, it actually waits till it has the presence of the host. So unfortunately, hosts leak chemicals through their roots, right? They leak chemicals into the air, so the pathogen's actually able to know that it's by its host. So it waits until there's enough water, then it germinates, and then starts moving towards its host. So one thing to note about the environment, why it's critical for that initial initiation of infection, once a plant is infected, the environmental conditions matter far less. So before I moved to Washington, I worked in Nevada, and we would have to do sudden oak death surveys because even though Nevada is extremely dry, if a plant coming in from another area where they have the right environmental conditions came in, it doesn't care that it's in dry, arid Las Vegas. It's already safe inside the plant, ready to keep this disease going. So moderate temperatures and moisture are important to this pathogen. So in our area in the Pacific Northwest, that means this pathogen is most active in about March through June and September through October. I remember 2017, Jay Scheidt was saying, oh man, this is a really good Phytophthora year because we had an extended period of cool, wet weather, and it was just perfect for these types of pathogens. That's why I have a nice flooding picture here. So, one caveat, environments can be artificial. Right? Just because our seasonal environment means that March through June is the best time for infection, if you are potentially creating your environment, so let's say that can be a field or in green industry where you're irrigating plants, you can then increase that ability for the environment to be conducive. So that could be based on how frequently you water, how often you water. And then for Phytophthora and these water molds like Pythium and downy mildew, they can sometimes also be found in recycled irrigation water. So they can move along in this water and spread around plant to plant. So environment can be natural as well as man-made or man-manipulated. Sometimes this is great because we can actually change how we treat the plants and water the plants to make that environment less favorable to the pathogen. So the history and distribution of this pest is it was first detected in the mid-1990s fairly simultaneously in both Europe and California. Now the true method of introduction is technically unknown. It is thought that potentially nursery trade could have played a role, but we don't truly know. Its current distribution, again, it is in Europe, as well as forests in California and Oregon and nurseries of California, Oregon, Washington, and BC. So Phytophthoramorum is a regulated pest. So that means destruction, and disposal protocols can be coordinated by the federal government and the state. So multiple times it has been found outside of the West. Sorry, give me one moment. I have to move my screen. Oh no. Okay. So APHIS may issue an emergency action order. So if we find this pathogen, then the diagnostician alerts their State Department of Ag and then USDA APHIS. And so then we go through trying to control this pathogen. So what happens is all susceptible plants, and this is what the previous order includes, this can change, all susceptible plants within 2 meters are destroyed. So that's a lot of plants, and the way they tend to be destroyed, and I think Marianne can comment on this, is we often dig a pit and burn them because that is the only way we can truly get rid of this amount of plants. And if you think about a nursery setting, there are a lot of plants within that 2 meters. And then all susceptible plants, so that's going to include those artificially susceptible as well, within 10 meters are functionally held in quarantine for 90 days and then made sure that they are free from Phytophthoramorum. So a massive response effort goes into this. Again, the USDA APHIS program, International Plant Management, Pest Management, Npdn, Forest Service, State Department of Ag, Cooperative Extension, and even Master Gardeners all team up to start tackling this problem with surveys, testing, as well as actually launching education campaigns. This is when we do public education. We talk about tracking and testing plants and why people need to be on the lookout for this particular problem. I have this poster that Washington Pest Watch put out because it shows just how many of us get involved when it comes to these pathogens. When we find something, we act fast. And that even means coming out with education campaigns to notify the public to like, hey, it's your responsibility too to be aware and be on the lookout and help us make sure this pathogen doesn't do more damage than it possibly can. So we talked about before, pathogens like to copy each other. There are a lot of lookalikes. I think the best way to explain this is think how many things cause a headache. You know, you could have had too little water, you could have been in a room with bad lighting for too long, you could be sick, this could be your 7th webinar of the day. You know, a lot of things can cause headaches. But figuring out the true cause is critical for diagnosis and for treatment. So lookalikes to be aware of include other pathogens, including other Phytophthora species, Low temperatures can cause cracking, wounding, bleeding. Sunburn, this is an ash in Las Vegas that was so sunburnt it started weeping. Abiotic root rot. Now, one of the conditions that Phytophthora like are kind of soggy soils. That can sometimes lead to just abiotic root rot. So those tissues just start sloughing off. Mechanical damage. I, Rachel, blight my trees quite frequently. Extreme weather, pests, things like chemical injury, fertilizer burn. The fact that rhododendrons just generally look suspect for pathogens is also one thing to note. So this is why just be involved with your diagnostician. A couple of lookalikes we have here. So this is some sunburn, sun damage on rhododendron. Now a trained eye can look at that and go, you know, that looks a little bit different than a perfect leaf blight, but you'll notice a fair bit of the sunburn is concentrated at that leaf tip. Here we have a Pyrrhus with potential lacebug infestation. And again, we see more tip necrosis. I will say best thing to do if you have any concern is you send it into a clinic. Best thing we can ever do as diagnosticians is tell you, no worries, it's just sunburn. You know, that's a great diagnosis for us to get to give too, but at least we know that you're looking and out there ready to catch sun note death. Other lookalikes can include sun scald, where we actually see a lot of bark damage, as well as cold injury. So if you were to find this tree and cut into it, this looks like a pretty sharp delineation from affected tissue to healthy. So it's concerning. And again, best thing to do is just send it into the clinic. It can be a really quick examination and test for us to say, no worries, not Phytophthoramorum. Verticillium wilt is another common one that we might find associated, particularly because of the flagging symptom. That's what we're seeing here, where only a few branches at a time actually show symptoms. And again, really easy to just send in a sample and let us examine for you and reassure you. So here's an example of another Phytophthora. So this particular maple, we saw rusting, splitting of the bark and the stem. So you go, oh no, this is perfect Phytophthoramorum symptoms. When we cut into the tissue, we once again see the discoloration, the sharp demarcation between healthy tissue and affected tissue. So what we had to do is we had to go down to the DNA level to determine that this was just Cactorum. This is one of those species, I don't even know if it's native or endemic, but it's been around and so present in so many of our landscapes that, you know, you kind of wipe your brow and go, phew, that was not Remorum, it was just Cactorum. So diagnosis, be aware of abiotic versus biotic symptoms. This can be difficult. This is another rhododendron just affected by abiotic, but it still mirrors some of those symptoms that we'd be concerned about. Trees with few feeder roots often can indicate that there's Phytophthora going on because it does like to move in through the roots. Bleeding cankers are always worth checking out, especially those rust-colored spots. Keep in mind the different tree species will have different responses. With leaf spots, check the tips and midveins as well as the undersides of the leaves. There are commercial field kits available. So that's a much blurrier picture than I was hoping for, but these are immunostrip tests where you can actually grind up some of that suspect tissue, stick this dipstick in, and if you have 2 lines, you know you have Phytophthora. If you have 1 line, then you know you have a negative result. So these can be very helpful if you are charged with inspecting a lot of plants. But again, we do need molecular techniques to confirm what that Phytophthora truly is. And obviously, if you find a positive, you should contact your diagnostician immediately. So see something, say something. If you're ever unsure of what to do or what you need, call your local clinic. On the Npdn website, we can direct you to where your most local clinic might be. And then the clinic can guide you on what type of samples you can take. They might suggest photos, or we're also really good at connecting you with the resource you need. So if you call me over in Eastern Washington, I can still put you in contact with other diagnosticians or researchers and solve that problem for you. That's kind of the beauty of the Npdn, is we're just here to really serve our first detectors and support diagnosis. You can also contact local extension, university, and state programs, as well as going to the First Detector website. Keep in mind, early detection is critical. Without early detection, we don't get that rapid response, and that's when we can see spread. So far, we've had really good luck. So reporting an invasive species, your state might have its own app. So the Washington Invasive Species app we have here, it's free, easy to use. It's powered by EdMaps, which a lot of different programs are. So that means data that we find actually goes into a national map. And for Washingtonians, it contains all of our noxious weeds. I have it on my phone for that because I need help identifying weeds quite frequently. Now, one downside of the Washington Invasive Species app is I do believe you have to come up with an EdMaps account. However, if you use our wonderful firstdetector.org website, you do not actually need to register with EdMaps to submit an invasive species report. So if you go to firstdetector.org and down here, you'll find on that third little box a report button. You click on the report and it takes you to a First Detector report form. So from here, you can actually search for a species based on what you found, where you are, and then you input your location and it uploads this information to EdMaps and then starts contacting the correct people within the network to help determine what that pathogen or pest might be. So there's a lot of times where we go, well, that doesn't really look quite like this pathogen. But again, as diagnosticians, as those of us who make these diagnoses, we love to actually see these reports because that means people are looking. And that means we will find these problems faster. Okay, so you're probably asking yourself at this point, does this really work? You know, we talk a lot about early detection, rapid response, and how it solves everything. Well, actually it does. Washington, we have a great success story. So we had someone who they took a training actually as a first detector, and they went back to the site that they managed, and they started to find that they had a rhododendron with some really suspect lesions. So they sent that sample over to Jenny Glass, and Jenny did some detective work, and she goes, well, that looks like Phytophthora. So she goes to Marianne, because Marianne's our queen of Phytophthora in Washington. She goes, Marianne, I need you to look at this. So then Marianne takes a look and goes, oh, I'm ad-libbing, I'm assuming she probably said something much more eloquent. And goes, we need to start contacting other people. So what happens is then they contact Washington State Department of Agriculture and USDA APHIS. So then everyone teams up, works together to confirm that, yes, this is Remorum. All of us agree this is Remorum. Now we need to go to the site. And from there, they start testing a bunch of plants in the surrounding area, the proximity. They close off the area to make sure that people aren't moving soil or water and potentially infesting other areas. They begin treatment. And then the cool thing about this site is the people who were at the site decided to make this a learning opportunity for their visitors as well. So they actually, once it became safe to do so and they knew that the issue was contained, they started advertising that they were doing research on this project, and that this is how devastating Phytophthoramorum could have been. Again, this is always that beautiful shining example of first detectors stop things from being worst case scenario. We can actually turn these into best case scenarios where, yeah, we found the pathogen, but now we're doing research, we've contained it, possibly eliminated it from a site. So if you guys have questions about that, Marianne is the one who's actually doing quite a bit of research at this area. But it is one of this shining example of this really does work. These trainings, these educations, and these partnerships that are built between green industry, diagnosticians, and then regulatory agencies really can save these areas. So submitting samples, because that is where diagnosticians really rely on green industry, because I don't get to leave the lab from about May till September. So submitting samples, whole plants whenever possible, we'll always tell you that, but that's not always possible, especially as we go into these larger landscape and forest plants. So choose portions of affected tissue, and they need to be usually about 6 to 12 inch portions with some woody tissue, some green leaves, and a range of symptoms. A totally dead plant is really hard to work with because a lot of other organisms have come in and started chewing on the tissue. As many photos as needed, most of us are walking around with really good cameras in our pockets. So take photos. This also helps document the symptoms. And let's say maybe you have a bunch of rhododendrons and then a pyrrhus next to it. If you send us a photo of that landscape, we go, I need you to also go and sample that lilac, that pyrrhus, that magnolia. And that way we can start testing to make sure that, you know, those plants are okay as well. Carefully package the plants, especially in the summer. And for those of you who are very good at growing plants, you want to add moisture or wet paper towel. We actually tell you not to do that because that can really speed up turning the sample into gush, and that's hard for us to work on as diagnostician. We also ask that you try and keep the soil off of the leaves and stems. So if you are able to send a whole plant, that means put the whole plant, the root ball, into one plastic bag, tape it off at the root line, and then put the entire thing into a second plastic bag. Keep the sample cool until delivery, whether you can put it into the mail or in person. Now, one caveat, if you are putting it through the mail, you need to treat it like a dirty diaper. Make sure that plant is packaged very, very carefully, multiple layers. I think we usually recommend 2 to 3 wrappings around the plant to make sure no soil nor leaf tissue can get out, or you can drop it off in person. And in general, with samples, delay equals decay. The longer that piece of plant tissue is no longer attached to the plant, the more rapidly it's going to decay. Most clinics will also have a sample submission form, so completely fill out that sample submission form. That saves everyone a lot of time because I'm not constantly, your diagnostician's not constantly having to call you asking you questions about, okay, but how do you water this plant? What soil is it in? What location did you find this plant? And then always remember, sometimes bigger is not always better. This was these were 2 giant blueberry shrubs that I got that outweighed me by a good 50 pounds. And I think I was only 2 inches taller then. And they actually put them through the mail. So, you know, call us first and we might be able to help you find the more appropriate sample size. So we do have a local resource. This is run by Marianne. This is our sudden oak death research and education program. You can find a lot of information about sudden oak death, Remorum, Blight. At WSU, we also have the Hort Sense page. It's written specifically for Washington, a lot of Western Washington-based issues and diseases. And with that, we'll take any questions. Appreciate so much time and energy you guys put in.
I don't know how you can tell what our energy level is, but I appreciate your energy level. It was great. Thanks, Rachel. Marianne, as people put questions into the Q&A box, I do see we have one, but do you want to add anything right off the bat to what Rachel covered, as give people a chance to put their questions in?
Well, I just want to talk a little bit more about that landscape situation that you were mentioning. It's a botanical garden in Western Washington, and they've done a really great job. I think Remorum had been there for quite a while before it was detected on a mature plant. It was a very large plant, you know, and it was all over. So it had obviously been there for quite a while before the worker had even seen it there. But yeah, they did a great job, and no more Remorum. You know, I think that they had all negative detections in their surveys after about a little bit more than a year. So that was really successful. You're muted. So
Sorry.
Sorry.
Well, I was going to ask, do you know, and there are some other questions, but do you know what the time frame was from when it was first detected through how long it took for them to completely eradicate it from the site?
Yeah, like I said, about a year. So they were doing, I think it was initially found like in March or winter kind of, and then everybody went out there in March, and they were doing like monthly surveys in the 2 areas that were really where the positives were. And then I think there was probably about a year or so of that. Then it went down to the usual twice a year survey that they do. And then I think if they're negative after 3 years of that, then they're considered to be free from Remorum. I just saw a question.
Yeah, so let me just mention this. So if you ladies would like to field the questions, the participants cannot see them. So can you read the question so everybody can understand what you're answering?
All right. Let's see. So there's one question. Does Remorum infect holly or Ilex? We have samples that look like the leaf damage you showed. I mean, there are other phytophthors that are found on hollies. I don't know if Remorum, if that holly is a very good host for it. I might be wrong though. We haven't found it on that host. Do you know anything about that, Rachel?
I'm trying to look it up right now.
Okay.
Well, this might be a great opportunity to say that USDA maintains the official regulated list of hosts, which they update as they...
Which I'm looking at right now.
Yeah.
So Ilex can be infested. It's plants associated with Phytophthoramorum, so it is possible. So just keep an eye out, but know that there are other Phytophtheras that could be associated in a lot of other diseases. But
And it's probably one of the other species on the holly.
yeah,
It's not like
it's really low down on the list, so that's a good sign for holly.
And let's see, another question, is there a best time of year to survey for symptoms? I would say no, because once the symptoms are there, you know, it's pretty persistent. There's a good time of year for them to get infected, which would be when there's a lot of moisture, like spring. But once the symptoms are there, I think you can see them pretty much any time. With Camellia, though, the leaves tend to fall off, so then you won't see anything on the plant per se, because the leaves have defoliated at that point, usually.
Can somebody answer the question from Ron? Are there treatment options? I think, Rachel, you did mention this, but if you want to explain that again, what happens?
Right now, if you do find Phytophthora, we're burning that plant. Let me specify Phytophthoramorum. That plant is destroyed. It's completely eradicated. There is no actual effective chemical control for Phytophthoramorum, and it's too risky to let that plant live. So getting a new plant will be the true management option. Now with other Phytophtheras, there are fungicides, primarily metalaxyl and mefenoxam-based fungicides, as well as, again, using that third side of the triangle of the environment, you can mitigate the impact of other Phytophthora diseases. Again, not Remorum, because if it's Remorum, we're getting rid of the plant. But you can reduce the amount of water it's getting or change its watering regime to reduce that period of soil saturation.
Yeah, and I just want to say about the fungicides, I think metalaxyl does work on Remorum, but because Remorum's quarantine organism, we're not messing around, we're just destroying it.
Fire is the management.
Yeah. Steam.
Steam, oh, that's right.
Oh,
I have some beautiful images of a thing that Marianne and Gary Chastagner came up with, where there are these steam boxes where they steam heat the material so that everything is just absolutely dead. And it's been so effective that we're actually using it in other parts of Washington to experiment with other quarantine or regulated pests.
yeah, like the vineyard snail, but I won't go into detail about
I know,
that.
it was like,
Well, don't bring up other scary things.
But,
Scary pest per webinar.
Other questions. Okay, there's some other ones in the chat. Simon asked if we detect Remorum in Washington streams. If so, what lineage is the dominant one in Washington? And we didn't really go into talking about the different clonal lineages. They're just like, how could you think about that? You know, we're all human beings, but we have different individuals. So Remorum, there are like 4 different clones or different individual types that are found. There's 2 European types and 2 North American types. And in Washington state, we have found both North American types and one of the European types in our nurseries and in our streams. So we have the three major types here. It's changed which one is dominant over the years. In California, they have primarily one of the North American types in their forest in Southern Oregon. Not so much the second North American and the European. We have all three in our nurseries and streams. So we're getting it from a lot of different directions, but it changes depending, during the years, which one's more dominant. I don't know if I answered your question or not. Let's see.
I relate them to donuts when I have to describe how strains work. It's like you have jelly donuts, maple logs, always food.
Lunchtime.
I always love your analogies.
Yeah.
They tend to be food related. There was a lot of grilled cheese today.
Yeah.
Okay.
It helps though. It's effective.
Treatment options for affected trees.
We covered that one, so.
Covered that one, okay.
Yeah.
The injection, did you talk? I wasn't paying attention. The trunk injections with the.
There are trunk injections, but I believe same thing, that trees getting removed.
Yeah, for Remorum. But I know in California they're doing some work with injecting the phosphite. Has hit or miss. But once a plant's infected, it's difficult to cure it. It's better to prevent, I think you were saying.
There's a question from Stephen.
Yeah, okay. How long do you have to wait to replant after an infected plant is removed? Our tree canopy is one of the notable characteristics. Keeping it intact is important. Okay, so where are you located?
I mean, I think one thing to keep in mind is you're going to be if you find Remorum, this isn't something you deal with on your own.
He's in California.
State Department of Ag, APHIS is probably coming in and helping you.
Yeah, and in California, especially in the Bay Area, it's fairly widespread. So I wouldn't replant with a host, like tanoak or oak or larch or anything. Maybe replant with something that's known to be a non-host for Remorum.
Well, and then there's the huge list of understory plants, right, which serve as reservoirs. So, I guess I would follow Marianne's advice and try to stay away from anything on that regulated host list, which I did add to the chat for people. Sharon had asked if I could share that link. So that is the link. You can also find it from the First Detector website, but I gave you the direct link. Anything else? Any other questions? I don't see any right now, so we're coming up on the hour. If you have any final questions or comments for Marianne and Rachel, now's your time to put them in there. I do want to say that we have some folks from the UK joining us today. So, here's a long one. But welcome from our friends overseas. I know you guys are battling with the same disease over there, really wiping out larch. Wow. Right, this is a long question, so if somebody wants to read that.
I'll read it. It says, I am the new nursery manager at a botanic garden in Central Coast, California. There's no best management practices implemented or sanitation protocols before me, and I want to start that here. I'm not sure where to start and don't think there are any records of previous tests done. I'm basically starting from scratch by cleaning up the nursery and pausing propagation. I think that's a great idea, and there are resources on the California oak mortality website about best management practices.
Oregon State also offers a online course through that Jennifer Parks program has put together it's really a certification course for nursery managers as well. And I think, as Marianne was saying, you can find it through that California Oak Mortality Task Force. I think I said that name wrong.
And it's very California. I mean, that's good because you're in California. And I think on our website a while back, we put some information for botanical gardens that we got from the UK, but very similar principles for nurseries and gardens apply. So I would start there by reading up on just trying to make some changes at your site to, minimize the damage. I don't know if Remorum is in the forest in the area where you are, but just try to keep it out. And if you have larch or anything like that in the gardens, those are good sentinel plants too, to tell you if Remorum is starting to come in. I don't know what else to say.
I will just mention, I don't know, Jessica, if you are a member of the Sentinel Plant Network, but the Sentinel Plant Network is a partner program with Npdn and the American Public Gardens Association. And members of that network, I see there's quite a few people whose names I recognize who are joining us today. That's an incredible network where people can share resources that they have. So you can reach out to me too if you want to find more information about the Sentinel Plant Network. Anything else? Last chance, folks. Right, I'm going to start the wrap up unless somebody has something that comes in the next 30 seconds.
If you have other questions, always just send us an email. I mean, that's kind of the beauty of, the Npdn First Detector Program and Sentinel Plant Network is everyone who's here is available and loves plants. So we're always willing to help you figure out, how to get the solutions you need.
Gotcha. I will say this, I thought of it when you were giving your talk. You mentioned the Npdn website. People can also look up their diagnostician now directly from the First Detector site. So again, whatever site you're most familiar with or most comfortable with, we're trying to just give you the resources and access to the information, in as many places as we're able to. So the First Detector's website is another place where you can look up that information. Great. Well, thank you, Rachel and Marianne, for a great presentation. I always enjoy working with you both on this topic, so I appreciate your time and expertise. Let's see, this is the last one of the series. We end with a big one, so thanks. All right, everybody, thanks for joining us today. I will, we are recording, and that link will be available on the First Detector training site once I get it converted. And yeah, thanks for joining us. Hope to see you guys next time around.
It was fun.
Thank you.
Yeah, it was fun. Thanks so much.
All right.